Hello, everyone. Can you hear me?
Yeah.
Yeah, great. Thank you. So I will start sharing my screen. Okay, can move this. Okay, let's get it started. Yeah, good afternoon, everyone. This week I will be talking about analysis of ChIP-seq data. Yeah, I'm sorry, but I don't have the fancy virtual background that Stefan has, so you have to be watching my boring walls and boring in my house. So yeah, unless I install some green, I paint everything in green, that'd be good. So but yeah, anyway, so last 2 weeks, Stefan already gave us a very comprehensive overview about some statistics and some models about the sequencing data analysis, as well as some very useful hands-on analysis tools for R and Unix. So my plan this week is to give you an overview about ChIP-seq data analysis. So this slide, this is my current plan for the 3 lectures in this week. So today, for lecture 1, I'll give you a brief overview about ChIP-seq technique and data analysis strategy. So the idea is to tell you that what part is important and what things you need to keep in mind when analyzing, processing and analyzing ChIP-seq data sets. And I'll talk about read mapping as the first step of the ChIP-seq data processing, and I give you an overview about the field data formats that are involved in the ChIP-seq data analysis. Because usually, no matter whether you start from raw data or processed data, you want to get an idea of what type of data it is, and what format it is, and what information we get from the data. And on Wednesday, for the second lecture, we'll be focusing on peak calling. It's the key part for ChIP-seq data analysis, and the main focusing with the focus on the common used algorithm MAX2 with the underlying statistical model that Stefan already talked about earlier today. And if time allows, I'll also mention another ChIP-seq peak calling tool, Sizer, which is focused on diffuse, like histone modification data. And we want to have an idea about quality control, which is a very important component in all genomic data analysis. So because usually always the case that if the data quality is not very good, if you follow all the analysis processes, for most of the time, you will still get most, if not all, the results. But how to interpret whether the results make sense, we have to give some measures about the quality to assess the quality of the data. And of course, data visualization using IGV, Integrative Genome Viewer, will be also important and straightforward to visualize and to display the data. And for on Friday, for lecture 3, I'll be introducing some possible downstream analysis and integration tools and several web-based resources. So after we get ChIP-seq data processed, after we obtain all the peaks, how to further analyze that to get the biologically meaningful information, how to interpret the data. So today, this lecture is I'll be talking about the ChIP-seq overview with these few sections about ChIP-seq technique, ChIP-seq data analysis strategy, And we'll talk for the second half, we'll talk about the sequence alignment and several things we want to pay attention during the first steps of data analysis and the data format. So ChIP-seq technique. Don't want this thing to overlay on the screen. So if I want to give a one sentence summary of what does ChIP-seq do? So ChIP-seq is to determine the locations in the genome that are associated with a target protein factor. So as I believe most of the audience, all of us know that in the eukaryotic genome, the genomic DNA sequences is wrapped, interacting with all different types of protein factors, protein molecules, and to form the structure of chromatin. And the several important protein factors that are associating or interacting with DNA, including histones. So histones form the structure of nucleosome, is the fundamental building block of the chromatin structure. And also other types of protein factors include transcription factors that can directly bind, directly interact with the DNA. And many of those protein factors will have a role directly or indirectly regulating gene expression or transcription program. So that's why we're interested in these protein factors that are interacting or associating with the genomic DNA. So we know most of the eukaryotic genome is extremely long, and for any type of protein factor, one important question we're interested in is where in the entire genome does this protein molecule goes, where does this protein molecule binds? So there are several sequencing-based, high-throughput sequencing-based techniques that can address this question. So ChIP-seq is, of course, one of the most important experimental technique to measure the genome-wide distribution of this of one protein factor. I also listed several other sequencing-based technologies, including RNA-seq, where we can directly measure the gene expression level, transcription level, for which Dr. Gurdon will be talking about next week. And DNA-seq and ATAC-seq are two technologies focusing on the accessible chromatin. So in which we're interested in where in the genome are the chromatin are open so that they are accessible to other interesting protein factors to come to. And Dr. Sheffield will be talking about ATAC-seq analysis the week after next. So for this week, we're focusing on ChIP-seq, in which we are, in this technique, we're targeting protein factors across the genome. That includes, primarily includes transcription factors or other proteins that are associated with DNA, as well as histone modifications or histone marks. So as I just mentioned, the target factors that ChIP-seq experiments can focus on are primarily they have to be DNA associating proteins. That includes transcription factors, which can directly interact or directly bind to DNA. And also histone marks include covalent modifications of histones and also histone variants. And also there are a category of important proteins called chromatin regulators that may or may not directly interacting with DNA. Most of them they are substrate, or they are interacting with other chromatin proteins, but not directly interacting with DNA. But they will have the function of like including histone modifying enzymes, including to write, to read, or to erase different histone modifications, and also some other protein regulators will have the function of regulating the chromatin structure, like chromatin remodeling complexes. For example, Swiss-Nif, or in human called BAF complex is an important group of them. So a very brief overview of what transcription factors do. So transcription factor is a type of a protein that have the function of regulating transcription eventually. So the structure of a transcription factor protein will usually have several components that they have the domain that they can bind by some ligands to activate the transcription activity, or they can interact with other proteins in order to activate this activity. And one of the most important functional domain in the transcription factor is the DNA binding domain, in which so this part of the in the protein that can directly recognize a specific DNA sequences, so that they can recognize and directly interacting or bind at that regions, certain regions in the genomic DNA. Usually they can recognize the specific sequences, and we'll mention it probably either on late Wednesday or on Friday to talk about the sequence motif scan. About once we identify the transcription factor binding sites, we're interested in looking for what specific sequences that this certain transcription factor will be targeting to. Another group of protein factors that ChIP-seq can help us study is histone marks. So histone, as we are familiar, that histone is the building block of a nucleosome. So there are 4 different types of core histones with 2 copies of each to form the nucleosome core particles, or some of the epigenetics field, we call this NCP. So this NCP does not stand for novel coronavirus pneumonia, which is the earlier name before COVID-19 was formally named. Anyway, so in NCP, there are 8 protein molecules with 2 copies of each of 2 copies of each of the 4 histones: H2A, H2B, H3, and H4. And H1 is another type of histone that help connect the different in between each individual nucleosomes. So the tail, usually the N-terminal tail of the histone molecules can be subject to covalent modifications that includes methylations, acetylations, phosphorylations, ubiquitinations, and a lot of other chemical modification that can modify certain amino acids on the protein sequences. And many of these histone covalent modifications will have some associations that can either change the local electric background or landscape in the local chromatin region, or have the association with gene regulation or gene expression. So I believe you are or should be familiar, but I'll just mention that to denote a histone modification, which sometimes we usually call it, say, H3K4 ME3, which refers to that is the histone modification on histone H3 at the 4th lysine, as a 4th amino acid, which is a lysine, and this is a tri-methylation, which means there are 3 methyl groups attached to them that modify them. So for histone modification. And besides histone modifications, there are another group of histone marks, like histone variants. So that's have a certain level of structure variations or protein sequence differences, differences on the protein sequence of the histones. Some commonly known histone variants include H2AZ and H3.3. So both of these histone variants have an important role of regulating gene expression, associating with regulatory regions in genome, and have associated with some function of gene regulation. And yeah, this is from a review paper quite a few years ago about what we knew about the functions of several common histone marks. So even today, these few histone marks are the most commonly studied histone modifications. And as we can see, most of them will have a regulatory function or gene regulatory function. Say K4 trimethylation are associated with gene promoter regions, and for H3K4 trimethylation and H3K27 trimethylation, if both of them co-localized in a gene promoter region, which we define as a bivalent domain or bivalent promoter, they usually mark the poised promoter, which means that this gene, although they are not expressed in this moment, that they can go to different expression patterns, either activated or repressed, depending on the gain or loss of either of these two histone modifications. And K36 trimethylation will mark transcribed gene body regions, and for enhancer regions, usually enhancer regions are marked by H3K4 monomethylation and also H3K27 acetylation. So also especially in the developmental stage, in the developmental stage, the poised developmental enhancers can be marked by K4 monomethylation and K27, H3K27 trimethylation. H3K27 trimethylation usually marked at gene silence chromatin regulated by polycomb repressed regions. And the H3K9 trimethylation are mostly likely associated with laminal associated domain and heterochromatins, also associated with silencing or inactive chromatin region. So next, I will give a cartoon picture or overview of how a ChIP-seq experiment is doing. So how to do it, what happens in the experimental procedure of ChIP-seq. So the first part of chromatin ChIP-seq for chromatin immunoprecipitation is for chromatin immunoprecipitation is that so for example, if we are interested in the genome-wide distribution of some protein factor, for example, this red star, so we can see in the entire genome there are a lot of different DNA associating elements. DNA elements that can specifically associate with a different type of proteins. Let's just assume that many of them are different transcription factors that they recognize different DNA sequence motifs. So in this experiment, if we are interested in the genome-wide locations of this red star, of this protein factor. So the first step is to do a formaldehyde cross-linking to tightly glue the protein and DNA, glue them together. And then we want to chop the DNA, chop the chromatin using either sonication, usually used for transcription factor, or a monocultural nuclease amylase digestion, which is usually used for histone marks, to chop this chromatin into small pieces of protein DNA complexes. So now the important step is to use a specific antibody that can specifically recognize targeting at that specific protein factor that we are interested in, we are targeting at. As we can see that this, they will also have some off-target effect that targeting some other part, some other proteins. So we use this specific antibody to target this, our interested protein DNA complexes as a process of immunoprecipitation, and to pull down this protein DNA complexes. And now we wash out all the protein part and only purify, only keep the DNA. So now we have the genomic DNA fragments that are associated with that protein factor we were interested in, we're targeting at. So the next step is to determine the sequences of the sequence of these DNA fragments, so that we can know where are these DNA fragments or the protein DNA interaction events, where do they happen across the genome. So that will involve several additional steps, including PCR amplification of these DNA fragments and high throughput sequencing. So yeah, so this is a overview of summary schematic of the entire process of a ChIP-seq experiments. Say for transcription factor, where the protein are interacting with certain regions in the DNA, and we use for either transcription factor or histone modification, we use antibodies to target the factor we're interested in, and to pull down, again, to purify the DNA and to sequence the DNA molecules or DNA fragments of these regions from the genome. So one thing I wanted to mention here is that although in the initial step, after pull down and DNA purification, all the DNA fragments we obtained, they can vary in length. And usually the sequencing, high throughput sequencing experiments, they cannot guarantee to get the whole sequence of the entire DNA fragments that we obtained here. That is determined by the next generation sequencing technology, or precisely Illumina technology. But in most of the cases, they can only guarantee to sequence one end of that of the DNA sequences. So usually, depending on what sequencing technique we use, so currently we can either sequence them single ends or paired end. In the case of in the single end sequencing case, what we obtained is the end sequence, randomly either end of the sequence from the DNA fragments. But nowadays, more and more people are starting to use paired end sequencing. Although, because in the case of a paired end sequencing, we can guarantee to get the information from both ends from the DNA fragments. But the cost will be, of course, the sequencing cost will be double, because we have both two sequencing reads information for one DNA sequences. So that's the whole procedure of ChIP-seq. So I'm always interested in some history about the development of some of these technologies. So I'll just give an overview of the history of how to develop this experimental technology. So the first cross-linking experiment was done in 1984, in which I think they were studying in bacteria in the case, but this is done led by John Lees. I believe Dr. Gerten is familiar with. Yeah. So this is from on the specific bacteria genes that for the first time they use cross-linking, but at that time they use ultraviolet UV cross-linking to study the protein DNA interaction. And later, for combining cross-linking with immunoprecipitation to study protein-DNA interaction, the first work was by a German group in 1993 and published in Cell. So that is the first work of using formaldehyde cross-linking chromatin plus immunoprecipitation. But at that time, of course, we didn't have much very powerful genomic technologies. So most of these biochemistry experiments, we can only study the certain parts, certain regions in the genome, or a specific type of protein DNA interaction without knowing the genomide information. So with the development of microarray technologies, which Stefan mentioned in a few lectures before, that so microarray technology it's a technology that they build up a lot of small oligonucleotides or small probes on a very small chip, so that they can, all of these probes can interacting or with the target, with some target sequences, target DNA sequences, that it can recognize whether your sample have this specific sequence or not. So people realize that by combining the microarray technology with the chromatin immunoprecipitation, we can study the protein DNA interaction across the genome in a genome-wide manner. So the first work that used chromatin immunoprecipitation with ChIP, combining with microarray, or they name it as ChIP-seq, was done by Richard Yang group, led by his postdoc Bing Ren in the year of 2000, and the work was published in Science. So a few years later, the next generation sequencing or Illumina sequencing techniques were developed. So people who studied use ChIP with ChIP-seq or combining with other technologies soon realized that next generation sequencing can be a powerful tool to combine with ChIP with immunoprecipitation to study these protein factors to give a genome-wide landscape or genome-wide profile of these protein factors. So in the year of 2007, two works were published independently led by Keji Zhao and by collaboration with Rick Myers and Barbara Rose, published in Cell and Science respectively. And they generated the first ChIP-seq data sets in the human genome, whereas the Zhao group sequenced nearly 30 different histone modifications plus several transcription factors, whereas the Myers and Wolf's paper, their work focusing more like a proof of principle experiment that generated just one ChIP-seq data sets with one transcription factor. So at that time, there was a competition, because once the sequencing technology was developed, it's fairly straightforward to think that to directly apply, to use that technology to study the genome-wide protein landscape. But then it's a matter of of course, it's a matter of a lot of experimental details to figure out the detailed protocols and to analyze the data. So, but in fact, the first work that used sequencing technology to study chromatin or to study nucleosomes were actually published earlier than that. This is a work published in Nature in early 2006, I think. Move this. no, earlier, 2 months earlier in 2007. But they studied led by the Frank Pugh's group at Penn State, and they use sequencing technology to sequence a lot of nucleosomes that contains the histone variants H2AZ in the yeast genome. So because yeast have compared to human, they have a much smaller genome. This sequencing depth can easily be very high. So in fact, this is the work by Frank Pugh using study H2AZ nucleosome genomide distribution of H2AZ in the yeast genome is actually the first work to use sequencing technology to study histones or any protein in the genomide. But in their work, they did not use the term of ChIP-seq. So that was a kind of unfortunate that although his work is published, is the earliest work, but if you use a matrix of a number of citations, which arguably is the right way to measure the impact of a paper. So unfortunately, the earliest work was not well recognized as the earliest work of ChIP-seq. Whereas in the competition between the Zhao group and Barbara Wo's work published in just 1 week apart. And this is widely recognized as the earliest ChIP-seq papers. And in that competition, there was another group led by the Broad Institute, led by Eric Lander and Bradley Bernstein. They were also in that competition, but they generated the first ChIP-seq data, including primarily histone modifications and RNA polymerase II. But this is the first ChIP-seq data in the mouse genome. But unfortunately, they're just a couple of months late. So but they are still recognized as the first work in the first ChIP-seq work done in the mouse. So they still received a lot of recognition. So just some history, some fun history about the technology development. So as Stefan mentioned earlier today, I was a graduate student in the Zhao group. Actually, this date, May the 17th of 2007, was the first day I worked in the Zhao lab. So I was fortunate to work in the lab in a very good time, to get a good opportunity to work on a lot of interesting things related to ChIP-seq. So of course, ChIP-seq has become a dominant method for profiling epigenomes or all the protein factors across the genome. So this is just the number of data sets that, of course, this is an incomplete collection of ChIP-seq data in human and mouse. You can see the number of data sets published in every year increases dramatically, increasing every year. And this is only, I think this only includes the data that are publicly available, deposited in the GEO, the Gene Expression Omnibus, but not including, there are more, few other repositories that have even more data sets. As for different histone modifications, we can see the trend of the number of data sets published, whereas in different histone modification. Because H3K4 trimethylation and H3K27 trimethylation are the two methylation marks that are most widely studied, that are most directly associated with active and repressive gene expression. So that's part of the reason these two modifications, these two histone modifications always have the most number of data sets generated by the community. As you can see, there is one factor, H3K27 acetylation, that have a rapid or very fast increasing trend. That although at the very beginning, people did not study this K27 acetylation that much, but one of the most important reason is because HVK27 acetylation is a very important enhancer mark that can distinguish the active enhancers from poised or primed enhancers. So, and the most transcription regulation events happens in the human or mouse genome occurred in enhancer regions. So there are a lot more acetylation, K27 acetylation ChIP-seq data that are being generated by the community. So yeah, this is some general review of the technology. And next, I'll be reviewing, give you a review about the strategy of ChIP-seq data analysis. I think, any questions from the audience? I don't know whether I have the, I think you can unmute yourself, right?
Yes, we can.
Yeah, thanks, Mike. Okay, so for data analysis.
Z?
Yes?
I've got two kind of worthwhile little quick comments.
Yes.
Alexander Varshavsky, for early ChIP, I would look him up. I know among, transcription factor folks, I think his papers are considered ultimately beginning to lock down modern ChIP. So he's just worth looking at. Alexander.
Sure, yeah.
I read chunky that paper from 1988.
Yeah, that is that.
Oh, I missed that.
Anyway, but it's also the kind of leads to modern ChIP. And then I just will kind of toot a horn for our paper. Mm-hmm. So we came right after Rick Young. Rick Young, his ChIP was a biased ChIP. In other words, they did it at promoters. So that was on a glass slide where you print the oligos. So at Affymetrix, we did it on genomic tiling arrays, which have the same unbiased character as ChIP-seq. And we did, in Tom's group, actually me and Simon Collier, first authors, we published the paper in Cell that was the first unbiased ChIP mapping.
Yeah.
That's notable. It's a citation classic. I just wanted to throw it out.
Yeah, definitely. Thanks, Stefan. Yes. So the genome tiling array is actually the real ChIP, microarray that can cover the entire genome. Whereas the first one, if you only cover the promoter region, that's only just a very limited parts in the entire genome. Yeah. Thanks, Stefan.
Can I make a comment too, Zhongzi?
Yes.
In terms of the single end versus paired end,
yes.
It used to be that it was twice as expensive, but you can if you talk to a sequencing facility, you can usually take a 75 base pair cartridge and get 100 reads out of that. So you can do a 45 base pair read and a 45 base pair read. And you might talk about this, for ChIP, you essentially, unless you're doing variation analysis, you don't really care. You want the minimum amount of sequence that will align to a genome. So you can do paired end read with a 100 base cartridge, because there's no such thing as a 50 base cartridge. This is what we started doing only about 8 months ago when we figured it out, but it's not too much more expensive. It's actually no more expensive, but anyways.
Oh, that's great. Good to know. Thanks, Mike. Yeah, so initially I was just intuitively thinking we have twice as much as sequence reads. I thought that probably cost would be double, but perhaps I can mention it a little more in the later lecture, but didn't prepare it today. That to determine whether you want to go with single end sequencing versus paired end sequencing, there are few actually more factors to take into account besides the cost. So depending on whether what's the biological question you are asking, what's the targets you're looking at. So there could be other factors to take into account in addition to cost. But that's a very good comment. Thanks, Mike. So a brief overview about the data analysis strategy of ChIP-seq. So as we know, we start from the sequences, the sequence reads that represents in the single-end sequence case represents randomly, almost randomly, either end of the entire DNA fragments that are associated with the protein factor we're interested in. So this is the raw data coming from the sequencing machine, which is in the format of a FASTQ that I will talk about later. And the first step to do is to align all of the sequence reads back to the genome. In most cases, we use the reference genome so that we can determine where is the genomic chromosomal locations of each and every of the reads comes from. So I forgot to mention that in the typical ChIP-seq experiments, the experiment is done from a large cell population of cells, which usually will involve in hundreds of thousands, at least 100,000, but usually perhaps in the early days of ChIP-seq, we require millions of cells, so that we can, because each cell will, in each cell nucleus will have 2 copies of DNA. So in that case, we can make sure that obtain the data that in the certain regions of the genome there can be multiple sequence reads that covered. So in this case, we can see this is just a random example from the genome that a lot of different reads, different sequence reads that are mapped in either strand, 5' or 5' strand or 3' strand of the genome. And we can see in this part of the genome, there are a lot of reads enriched here. So which could indicate that this part of the genome, the protein that we are interested in could interact there. We could have a binding sites or interaction. But this type of direct alignment is not, after we got the alignment, is visually is not that straightforward. It's not, certainly not enough to get a clear understanding of what the data look like. Then we will see, we'll convert this type of directly reads alignment to this type of profile that we can browse in the genome browser. And on top of that, we will use peak calling algorithms or peak calling tools to identify where in the genome are they actually peaks, or the regions that have a statistically significant enrichment of sequence reads compared to the control or the random background, the background across the genome. Also, different factors or transcription factors or different histone modifications will have a different pattern as shown in this genome-wide profile. This is an example of a typical transcription factor, what the data look like, and what a histone modification could look like. So you can see that for transcription factors, it's pretty much fairly easy to see clearly to see which the peak is. But for histone modification, it could be more trickier, as the data could look very noisy or diffuse, or they form instead of forming a sharp peak, they could form a broad peak, making the peak calling trickier. So by saying that, we want to do this, namely these few steps in the data analysis. So first step, we want to address the question that where in the genome do these sequence reads come from? By doing that, we'll use sequence alignment to address that. And the next question, where does the enrichment of sequence mean? So we will use peak calling to identify the regions that have the enrichment of sequences. And the third step is what can we learn from this data, how to interpret this data that will involve in some downstream analysis and integration. So, so for today's lecture, we'll primarily only be able to cover the first part of this work. So let's get to revisit the entire experimental procedure and the computational or bioinformatics analysis procedure of ChIP-seq again. So the thing I wanted to point I wanted to make is that in the experiments, we follow several steps of a chip, antibody pull down and get the DNA, and doing the library preparation and do the sequencing. And the procedure of data analysis of ChIP-seq data is can be regarded as a reverse process from the raw sequences. It's a reverse process of getting back to each step by step. We will get back to the original biology or original pattern, original picture in the cell nucleus. So in details, so when we got to the raw sequence reads, so this is the raw sequence reads are the raw data that we start with in terms of ChIP-seq data analysis or analysis of any next-gen sequencing data. So, and the raw sequence reads is usually in the format of a FASTQ. And we do sequence alignment using a tool called Bowtie 2, among other tools, of course, to get the mapped reads. So difference between the raw sequence read and mapped reads is that the mapped reads, we will know the genomic location, the genomic coordinates of each of the sequence reads we obtained from here. That represents where in the genome, not only from the random DNA fragments, but we know the information of where does these DNA fragments come from in the genome. And the quality of the raw sequence reads and the alignment will represent the quality of including the sequencing procedure and the library preparation procedure. And then after mapping the reads, we will assess the redundancy of all the mapped reads. I will talk about a little more detail later. And the actual informative sequence reads are the non-redundant reads. And this step of quality assessment is kind of assessing the quality in the library preparation, process. Namely, the redundancy assessment can give an idea of how likely there was a PCR overamplification of the sequence of the DNA fragments. So if the starting material is not enough, certainly you can just undergo more cycles of the PCR amplification to get more sequence reads to fit into the sequencing machine. But more DNA, more sequences doesn't mean we get more information. So that's the redundancy assessment step is talking about. So with the non-redundant reads that represents the DNA fragments that directly coming from the ChIP experiment, we will do peak calling, identify the regions from the genome where the protein-DNA interaction occurs. That in this step we will capture the actual biology information of where in the genome do we have the signal, we have the information. That is for peak calling. Besides that, of course, we will also do piled up, just to pile up all the DNA fragments represented by the non-redundant reads for visualization with the data formats mentioned here. And all of these steps, from the mapped reads to all the redundancy assessment, peak calling, and pile up for visualization, all of these steps can be done by bioinformatics tool Max2. So after, also we'll be mentioning about the QC quality assessment in this step. So after we are obtaining the peaks that represents the protein DNA, the regions of protein DNA interactions, if the data, the quality of this step, the quality assessment of this step will be able to tell whether the peak calling step will be able to tell whether the ChIP experiments worked well or not. So if the chip or the antibody or also the quality of the antibodies, if the antibody is very efficient and you did the experiment very well, you can obtain a lot of capture a lot of protein DNA interactions that will obtain a lot of peaks in the genome. So this is having the assumption that in the genome as big as a human or mouse, in any histone modification or any transcription factor, there are usually at least thousands of protein peaks that can be identified, or thousands of locations in the genome that have this protein DNA interactions. So usually it can be more, and in rare cases it can be fewer. So having this peak identified, the next step is to how to integrate this information, and then we'll use downstream analysis and data integration to integrate the data and to get the biology information from there. So now I'll briefly mention the first step after we got the raw sequencing data from the sequence machine. We have the FASTQ raw sequence reads. We can usually use a algorithm called FASTQ-seq to briefly assess the quality of the sequencing of the sequencing runs. So FASTQ-seq is a very straightforward algorithm that is already, I think it's already pre-installed on Grafana, that you can do module load and run FASTQ-seq on the FASTQ raw sequence data file, and they will tell you a global they will give a series of different QC measures in the format of a HTML website format that you can easily browse. So some of the useful quality assessment, including the quality scores of all bases. So this represents the position. So this is a position in each of the sequence reads. So this is the 50 or 51. Each sequence reads will have 51 nucleotides, and this is a distribution of the quality score of all the bases in the first base of all the reads and the second base of all the reads. And you can see this is the quality score distribution along this score. The green in the green area means that the quality is good, and I mean, if you're down to the red area, it makes that this is questionable. This is the quality is not very good. So these are two examples, the examples of two ChIP-seq data sets, two real ChIP-seq data sets. So we can see in this one, all of the in every base pair across the entire sequences, they have a pretty good quality score. And whereas in this one, only the first few bases have an okay quality score, but then the quality dropped down dramatically, especially after 25 base pairs, meaning that perhaps when we read the sequences, everything after the 25th bases may not be as trustworthy or as reliable as the reads in the beginning. Another useful sequencing quality assessment that I can obtain from FASTQ-seq is the sequence content across all bases, which means that in each basis, again, across averaging across all the short sequence reads from the data sets, we look at the first basis, and this tells us the percentage, the fractions or percentage of the 4 different nucleotides, A, T, C, A, and G, the percentage of these 4 nucleotides distributed in the first basis, second basis, and so on and so forth. So this means that we can see across the entire sequences, from the first base to the 50th base, the distribution of the 4 bases are fairly very stable. This is a good data set, meaning that it really captures the the real DNA sequences from the genome, which usually represents a relatively stable distribution of the GC and AT contents. And as well, in this case, this is another example. We can see the distribution are very messy, but something we can read from this sequence content plot is that the first base we can see 100% is 100% T. The second base 100% A. So from the first few bases, you can actually read the sequence. So that's a T, A, C, A, G, A, and G. So which means that no matter which sequence reads you are reading, among the usually tens of millions of sequence reads, the first several bases are always have the same sequences. This usually indicates that's a bad sequencing quality. That usually it could be the case that there are certain something happened in the library preparation, in the experimental procedure, or in the that the sequencing machine actually captured the possibly the adapter or several other sequences that are unrelated to the actual genomic sequences. Any question?
I had a practical question and I don't know the answer to it. So when you get like a fast QC that has like a bad read, like the previous one, like from position 25 onward, do you just trim those or ignore those, or what do you do from like a practical point of view? Like, do you just throw away that data or what?
So first we simply ignore this QC measurement, but just to do the sequencing alignment to see to look at the quality assessment of the sequencing alignment. Even if some of the sequencing quality, some of the bases, the quality are bad, but in that case, even in that case, as long as most of your sequence reads can still be mapped to the reference genome, that means that this data sets, the sample, the data set is still usable. So then I will probably just simply ignore this fast QC results, but just to go ahead with whatever we got. But this is usually useful in the case that you have fairly few amount of sequence reads, or the overall sequencing, you only get a very few percents of reads that can map to the genome. Very few, I mean, say less than 10%. So if you sequence 100 million reads, you only get less than 10 million reads that can map to the genome. That could usually tell that there's something went wrong in the sequencing step, that we want to consider trim certain reads from that and try it again. So if this is happens, well, for the sequencing quality score, but in this case, if this happens, usually none of the sequence reads or very few of the sequences you can actually map back to the genome. And from this type, you can clearly see. And in this case, we will have to trim the reads to do the mapping.
Yeah, that looks like a barcode, right? That someone had.
Yeah, it looks like a barcode or some adaptive sequences. Yeah.
Yeah, we do the same thing that you do on the previous, which is essentially ignore the fast QC and align to a genome. I just wanted to see if that was the correct way or the way people typically do it.
Yeah, so once we see it, certainly there's no way that this barcode or alignment or barcode or adapter sequence can map to the genome. We'll have to either trim or redo the experiment. Yeah, so that's yes, question?
I have question. So as I well understood, in normal condition, Illumina doesn't sequencing the adapters, because on the left picture, we don't see the first 6 base pairs.
Right.
So it means Illumina doesn't read, during the sequencing, Illumina doesn't read the adapters.
Right, which is supposed to be the case. So the sequencing machine is not supposed to read to sequence the adapters. So this step has to be taken care of in the library preparation or some step, so that to make sure that whatever the sequence reads that fit into the sequencing machine, that the machine sequence should already exclude the barcode or adapters. Well,
okay.
I mean, for barcode, that could be a different story, because you probably want to, well, that's not the case. Even if you include the barcode in one sequencing run, you should include more than one barcode, right? Not only the same barcode, right? Am I?
Yes. Thank you.
Yeah, if the entire full cell or the entire sequencing run contains only one barcode, that still means that likely this experiment, the sequencing failed, because you don't get multiple barcode information. So in terms of the time, okay, we will briefly introduce so the next step will be sequence alignment, which we want to align all the sequencing reads back to the genome. And we'll give a brief introduction of the underlying key algorithm of most of the sequence alignment algorithms, including bowtie and BWA. We can see they both, the name of the algorithm both have a B and W here. They all refer to Burrows-Wheeler transform, which is an efficient transformation algorithm to process or to index the genome, so that they can make this sequence alignment or read mapping process extremely faster than you compare each and every of the tens of or hundreds of millions of reads back to to align each read to the 3 billion base pair genome. So Burrows-Wheeler transform is originally developed for data compression. So it's a reversible permutation of the characters. So I will use this as an example to show what the Burrows-Wheeler transform is. So for example, if we have a sequence, if we have a string of characters, in our case, it's ATGC, nucleotide sequences of A, C, AA, CG. And then after this, we add a $sign to make that as an end of string, like a coda, like a $sign. So the Burrows-Wheeler transform is to first what we do is to rotate this sequence. So say it's an AC, AA, CG $sign. That's the original one, and then we move the first one to the last, but keep all of the sequences unchanged, and get to the one rotation transform of the sequence, and include all the possible rotations in this string. So as we can see in this, all of the rotations, the sequences the one next to each other is always keep the same, keep unchanged. Say this A is followed by a C. So in every sequences, this A should always followed by that C. So we get all the possible rotations to generate this matrix. And then we sort the first column. And then the Burrows-Wheeler transform is simply record the last column as the transformed case of this one. So after resorting, this matrix is called a Burrows-Wheeler matrix or BW matrix. And to keep so instead of storing the original string, we store the BW transformed string. And this string is an intuitive understanding of this transform is that this string contains both information. One piece of information is the which character is after which character, the relations of all the characters. They somehow contains in this matrix. And the second piece of information it still contains is there's some kind of a sorting information. because as long when you compress the data, you want to when you compress the data, the more sorting you did, the easier you can compress. You can compress all the if you sort if there's a tie, you can compress all of the same thing. But the sorting process will disrupt or will disrupt the information of which character is after other. But this process keeps that information somehow in that way. So by using this Boris-Wheeler transform to index the entire human genome, the bow-tie and BWA algorithms, they can make the sequence mapping procedure is much faster. So I won't go to the details of that algorithm, but I thought that will be useful or to know, to get a brief idea of what BW transform is about. So for sequence read mapping using bow-tie or BWA, our strategy is that we only want to keep the sequence reads that can uniquely mapped to a certain unique location back in the genome. So that's coming back to the early days of the sequencing experiments. So we determine for each sequence reads, if they cannot map to the reference genome, of course, we will have to discard these reads. There could be several reasons. Most likely, there's something wrong happens in the sequencing or the library preparation or sequencing step. Say they include a few base pairs of a few bases of the adapter or other information so that they can no longer align. Or very few cases, it's possible that there are some. I mean, the sequencing could be wrong. They only allow 1 or 0 mismatches with the reference genome, but the reference genome may or may not be exactly the same in every single basis with the actual genome you are studying. But this is a very rare cases. Most of the cases will happen in the sequencing error. And if the reads can map to multiple loci and multiple places in the genome, in the early days, we simply discard this type of reads because we cannot determine the exact unique location in the genome. That makes the other studies difficult. But recently, as long as the sequence quality is still high, I think both bow-tie and BWA can still keep this information as a score of giving some weight and map to all these locations in the genome. But this is also not an extremely essential point in this part. But the most important is that we keep the sequence reads that can uniquely map to that one location from the reference genome. And the next thing I want to talk about is the redundancy assessment. So because all of the DNA fragments is coming from either sonication or a micrococcus nucleus digestion that chop the chromatin and the DNA into fragments of DNA. So most likely, also the cells are the sample DNA are from a large population of cells. If we compare different cells, for either amylase digestion or sonication, say a transcription factor molecule or a histone or nucleosome, they will associate where this part of the sequence is usually at least 50 or usually more than 100 or 200 base pairs. And the cut location here, the cleavage location here across different cells is very rarely can be the exact base pair, exact location across the genome. So for the same regions in the genome, for the same proteins in the genome, if we have a very strong TF binding, as this one example, so which means that we can get a lot of DNA fragments from this region. But the sequence reads on both ends, most of them, their coordinates could be a few base pairs apart from each other, but not the case of the exact same location. So in this case, we frequently from the raw sequence data, so all of these reads can uniquely map to that exact same location in the same genome. But this case, there could be a lot of reads mapped to the same position. So for this case, in this case, most likely these are due to the overamplification in the PCR or library preparation step, but not represents the real information. So this can be more detailedly addressed by some statistical modeling to see how likely are two reads mapped to the exact same location. But I mean, simply speaking, usually in the ChIP-seq data that have 10 to 20 million reads, this type of events is very, very rare. So even if we remove them to keep only one copy of this case, we won't change the actual biological information. So this is what we call the redundant reads. So we only want to keep the non-redundant reads from that from the sequence reads, from the data. Okay, next I will quickly go through the several data formats that will be involved in the ChIP-seq data analysis. So first is the raw sequence data file in the FASTQ format. So FASTQ format is actually a pure text format. Although usually it's a gzipped, you can unzip and use any command to open it in or even a text editor to open this file to look at the file. So you can see each sequence reads is represented by 4 lines in the FASTQ file, and the first and third lines are just a identifier or a name of the sequence, which doesn't mean much in the analysis. And the second line represents the actual sequence of this sequence reads, and ATGC and N means and determined. And the fourth line is the sequence quality score of each of these sequence. And the quality score is coded by the ASCII code. So some code will have a lower score, some code will have a higher score based on the ASCII code. So 4 lines represents 1 sequence reads. And in this case, an easy way to measure or to calculate how many total reads from the FASTQ data file, you can simply use WC word count from the WC-L, the line count from a Unix command, and divided by the total number of lines, divided by 4 to get the total number of reads from the FASTQ data file. And another most commonly used data format is BED, BED format, and usually used in the after sequence alignment, after aligning the sequence back to the genome. And it's also easy, very straightforward to interpret and to read. That includes the first 3 columns in that data is the most important, represents the chromosome, calling which chromosome that this mapped reads coming from, and the start coordinates and the end coordinates. So having this first 3 columns, we can uniquely identify an exact location in the genome. In any well, of course, we need to define which genome it is, human or mouse. But this information can represent either the mapped location of a sequence reads, or any regions in the genome, or even later on we'll see a peak we identified from the genome. And the fourth line, the fourth column is actually a name or identifier, which doesn't really matter, it doesn't really mean anything. And if the fifth column is a score, sometimes the score is useful, it's meaningful, and sometimes it's not. So in this case, it didn't store any useful information, but keep it as a spaceholder here. And the sixth column represents the strand, the directionality information, whether it's in the positive 5' strand or 3' strand in the genome. When the sequence reads, well, the strand information is usually not meaningful for peak region or any region in the genome. But as for a mapped location of a specific sequence reads, whether that's a positive strand or negative strand is extremely important. And there are some extended byte format that could add additional column or additional information that you can simply look for them from a website, from online. with saying what that is. And some other data formats that are used are SAM and BAM that usually are used by Bowtie and BAA to store the aligned sequence reads. And SAM is also a readable data format, and BAM is the binary compressed format of a SAM format file that it contains both the original sequences and the alignment coordinates, alignment locations of the information. And several other data formats that we'll briefly talk about, and then we'll mention in more details in later talks, later lectures, including bed graph. So bed graph and Wiggle and Big Wiggle is to build up a profile for browser visualization. So bed graph file is also a variant of a bed file in which the first three columns is still chromosome start and end. Sorry, I think I may have said it wrong. So the fourth column should have been the score, and the fifth column is the name identifier, which can be random identifier. The fourth column. So for bed graph format, they only have the first four columns as a bed format, and the fourth column is a score that represents the height, or whatever the score of this region. So as you can imagine, if we just tied up all the regions across the genome, or every region that have a signal or have a sequence reads, we label this start and end location with a score represents as a height that can represent that can generate such a profile. And Wiggle is just a more compressed format than bed graph that requires this all the windows, all the bins are equal length. And the Big Wig is also a binary, really a binary compressed format of a Wiggle format. All of these are used for visualization purposes. So I think we'll just end here for now. Sorry, I have already gone over the time. I'll just revisit and summarize this entire strategy of the data analysis for ChIP-seq data. And as this represents a reverse procedure of retrieve or trace back each step from the experiment, the actual experiment. And in each step, I want to emphasize that quality control is extremely important, as well as whatever we got using any tool, any methods, we need to make sure that the data actually works well. I mean, the quality is worked well, quality is good, so that any of the later results we receive will actually represent some biological meaningful information rather than random noise. Questions?
Can I ask a stupid question? So I was just wondering, how do you differentiate between a PCR amplification of certain sequence with the height of the peak in a certain region? Do you understand what I'm saying? Yeah.
So that's a great question. So in the redundancy assessment step, simply speaking, if all the sequence reads have the exact same start and end and strand information, most likely it is PCR overamplification. For real biologically meaningful reads that are enriched in a certain region, they usually have different start and end locations. So even if all of these are together, so maybe I can use this one as a schematic as an example. You can see if the sequence reads are enriched in a certain region in the genome, many of them should have a slightly different, very different start and end location. So that after piled up, we can form a mountain and form a peak-shaped profile. Whereas if all the reads, a number of reads have the exact same location, same start and end location, after piled up, they will be just a spike. a very narrow spike that doesn't have the shape of a mountain shape or triangle shape. That is another way of distinguishing them. But in terms of data analysis, as long as the sequence reads have the exact same and the same location, same start and end location, we'll just discard them as, well, only keep one copy of them as calling the rest of them are redundant.
Yes, I understand. Thank you.
Sure.
And what is the average read depth you generally target the in ChIP-seq? Is there a
You mean average read depth?
like the total number of reads you target usually? I mean, there is certain limit for RNA-seq, right? There's so many reads are good for quantification. Is there something?
Oh yes. So usually in the read depths where it means that the number of informative reads, if I may. So that in that case, I will always go with the number of non-redundant reads in the data sets. So which means that we have discarded reads that cannot map to the genome, and also discard all the redundant reads that do not give us any additional information from the data. So all the non-redundant reads, number of non-redundant reads represents the informative or the actual useful sequencing depth of a ChIP-seq data set. Make sense?
Yeah, thank you.
Okay. More questions? So yeah, I've posted the assignment documents in the Collab site, and you can start browsing them and let me know for any questions, and we can go all over some of these in the next lecture. Okay. Okay, and I think I have. Let's see. I believe you can find my email, and let me know if you have any questions anytime. Otherwise, I will see you on Wednesday.
Thank you.
Okay, thank you very much.
Thank you.
Thank you.
Thanks, Chengzi.